Good morning and welcome to the third in the series of phonocardiography. Today what we're going to be doing is applying the phonocardiography into human performance, which means basically looking at the production of energy and how to enhance the energy production and factors which inhibit energy production, both in non-sporty people as well as sporty people. So the first thing I want to just run through is just to say what we're going to try and cover today. Now, we don't know as far as time that we'll cover every single topic, but we'll do our best. So we want to review a little bit about the phonocardiograph itself. We want to then look at the challenge for the emotions, and particularly the subconscious emotions that can cause blocks in our performance. So we're simply going to do the techniques to balance the conscious and the subconscious. And then we're going to be looking at body types of the gene expression, or the genotype and the phenotype, how the body's expressing those genes at the moment. And then we'll review the eye patterns, eyes into distortion. Then we'll look at the production of energy and how to challenge for ATP in the mitochondria, and also for hypoxia, which is a lack of oxygen, or the fuel, the air, if you like, within the furnace of the mitochondria. Then we'll look at the different nutrients that are necessary for the production of energy and the different aspects of what can create hypoxia and how to solve those problems. We'll be looking at briefly about toxins, particularly in relationship to toxins inhibiting enzyme functions, toxic metals, radiation, chemicals, the role of infections likewise, and then the difference in the energy requirements when somebody is out on the track or exercising aerobically or anaerobically. Okay, so let's quickly review the phonocardiography. One of the things that we're always asked is about stethoscopes. Do you actually have to have a digital stethoscope to do phonocardiography? And the answer is no, you don't. Okay, you can use a perfectly ordinary, regular stethoscope. The only difference is, of course, that with an ordinary stethoscope, you're not going to see the graphics on the computer, but you'll get the same readings back using the muscle testing with the patient as you will with the digital one. So the advantage of the digital one is you get the recordings, and then the nice thing is you can then play that back in next time the patient comes, in a month or in 3 months' time, and say, this is what we were, and this is what you are now, and you can see the difference. And hopefully the difference is better rather than worse. Okay, we were told over last weekend, 2 weekends ago at the ICAK conference in Washington, that the Canadian ice hockey team is now taking this up using phonocardiography to assess the performance of their players. So these are professional players. So it's beginning to get into sports world, which is great, as an objective way of monitoring human performance. So phonocardiography is not a new topic. As far as I'm aware, historically, it came out of the works of Roy Lee in the 1940s with a rather big box and a wax paper and a needle, which used to operate, and then the doctors would look at this. It was revised in about 1980s into a more system-like, more like a fax roll with some form of play out. But what I wanted to do was to be able to actually muscle test people using the phonocardiography. So the idea of using a digital stethoscope made sense. And then once I realized that this actually worked using kinesiology, you could use an ordinary stethoscope in the same way. So basically, the revolutionary part is we do the recording of the patient's heart sounds, and then we play them back to the patient. And we bypass the lower parts of the brain, the hindbrain and the midbrain, by putting it straight into the temporal lobes of the cortex. And then it's relayed from there into the frontal cortex and parietal lobes for analysis. So what we're doing is we're bypassing the parts which filter out the noises that the body hears all the time. Because your body doesn't hear your heart, even though it's not too far away from the heart, from the brain. It doesn't actually hear or feel it because we've got acclimatized to it over all the years of our life. So the cardiography, the difference is that the feedback gives us a way of evaluating what it is that the patient needs and what is the cause of their problem. Now, I don't know the cause of people's problems. We have a general sort of rule that if this particular shape shows on the graph, then you might want to think about vitamin B, vitamin B2, vitamin B12, and so on. But nothing is as precise as that, as we know with humans. You have to test. And the great thing about using the kinesiology is you can test precisely what nutrient or detoxification remedy a person needs and the dose. So phonocardiography is a diagnostic technique, but we've applied it into actually not therapy, but to apply that much more functionally by feeding it back. It can be performed by a digital stethoscope, but similar results are achieved by a standard one. Now, what we learnt between the first and the second seminar, between November and March here that we did, is we were able to tie in the 3 body types, the different builds with people, and the different cardiac performance or ailments that they suffer from. In the first one, we talked about people who needed to vasoconstrict and people who needed to vasodilate, and that was the difference between the different forms of vitamin B. So vitamin B1 was a constrictor of the blood vessels, and vitamin B2 and 3, which the Americans call vitamin G, were dilators. And then we met people who seemed to weaken to homocysteine and were more subject to cardiovascular problems and cerebrovascular strokes. But it wasn't for a little bit longer before we realized this was actually the body types. And we are familiar with the red constitutional body type of person, whose the middle height tends to normally be on the thinner build and are governed by their thyroid. But they do have the ApoE4 expression, which means that they can't detoxify cholesterol fragments very well, and metals, and particularly the metal that they have a strong affinity for, red people, is aluminium. So never forget this basic material is that red people are blotting papers for aluminium, and aluminium can be a poison to inhibit enzyme pathways. These people also have high homocysteine because there's an enzyme called cystathione synthase enzyme which converts homocysteine into cysteine, which is inhibited or not expressing itself with the allele very well in red people. So this allows these people to have high levels of homocysteine, which may inhibit the formation of collagen to some extent in their blood vessels and tissues. So red constitutional people have high homocysteine. So the homocysteine formula that we made, we just extended that out to a range of vitamins and minerals and coenzymes suitable for a red person. So we now do a red mix, as you're familiar with, because red people have this predisposition towards cardiovascular problems. Green people, we met, are the taller people. They tend to be on the bigger side and live on their adrenal glands. And green people, we noticed, were not the people who got the heart attacks so much, although they can get heart attacks, but they got the hardening of the arteries. This was the atherosclerosis. So green people, therefore, are people who need vasodilating. Their tendency towards hypertension tend towards constriction because they live on their adrenals. They have high levels of aldosterone, etc. So this was much more what the Americans call vitamin G, or relaxing, dilating factors. So green people tend to need G for green, that's the easy way to remember it. They need vasodilating, so they need the B2 and the B3 and the other associated vitamins with that, like B6 and folic acid. Greens also need to keep their cholesterols down because they have a tendency towards high cholesterol and atherosclerosis. So we put into that mix choline and inositol and biotin, the lipotrophic factors that help us to break triglycerides down and keep them moving. So the last ones are the blue constitutional types, and what we found is that blue people tend to be on the shorter side. For females, 5'5" or below, maybe 5'6" for a man. But these are the ones who have the electrical problems, the firing sequences, the wiring within the heart itself. So the heart variation in the waves, the electrics, they're the ones who get the ectopic beats, the missed beats, the heart blocks, the bundle branch blocks, etc. So these people tend to need the B1. The B1 aids the conduction or stimulates the production of electricity in the mitochondria and the neurons, and also B4, which was adenosine, which is rich in wholemeal bread and yeast residues. So the blue people have the electrical problems, okay, the arrhythmias, etc. The green people get the hardening of the arteries and need the vasodilation with the B2 and B3 factors. And the red people are the ones who need the homocysteine of the B6 or pyridoxal 5-phosphate and adenylyl, rather methylcobalamin and folic acid and the other nutrients involved with that. So this is what we found between Module 1 and Module 2. Now what we learned in Module 1 right at the beginning is that when you listen to a heart with a stethoscope, you hear two sounds. When you feel a pulse, you only feel one pulsation. Okay, that's the pulse or the pressure wave coming from the heart. But when you listen with the stethoscope, you hear two sounds, and these are technically or correctly called lub and dub. And lub is the first sound, and dub is the second. And then we should hear lub dub again and lub dub again. And between each lub to the next lub is called a cardiac cycle. And it is roughly 0.83 of a second. So all this happens, the whole cardiac cycle happens in less than a second if the pulse rate is about 70 times a minute. What we learned is that the lub-dub, the second sound here, is smaller than the first sound. And that the first sound should be, in an ideal situation, 2 to 3 times higher than the second sound. Okay, now we can't measure it from the cardiograph precisely how efficient the heart is, because some people's hearts are set further back and so the sound can be quite quiet. It's the ratio or the amplitude of the beat which is important. So it's the ratio of the first to the second sound. So the first sound should be 2 to 3 times the height of the second sound in an ideal. We very rarely find that, as we found. The distance between the second sound and the next first sound should be twice the length of the first sound to the second sound. Should be twice the length. And the reason for this is that between the first sound and the second sound is when the heart contracts. Basically, when the ventricle contracts is the working time of the heart. And we all, if you remember, what we did is we clenched our fists and then we relaxed them. Clenched our fists and relaxed them. And the myocardium, or the muscle of the heart, has capillaries that go down eventually from the coronary vessels to every single fibre in the heart is supplied by a capillary. So when the muscle fibre is contracted, there's no blood flow to the tissue itself because it constricts it. So it stops the blood flow of oxygen and the nutrients to the myocardium itself. So it's a danger if the length of time of the relaxation of the heart is shorter, because it means the heart itself won't get the blood supply. So with the phonocardiograph, we look for the distance between S2, as it's called, or dub, to the next one, and that should be twice the length. If it's getting shorter, this means the heart isn't relaxing enough, and if it gets to halfway, in other words, the same size, the same length as this one, there's a distinct risk of a coronary attack. So when cardiologists have been asked if an electrocardiograph, which measures the electrical charges in the heart, can predict a heart attack or myocardial infarct, the answer is no, it can't. It can tell you after it's happened, which isn't much good to most people to say that you've had a heart attack, but the phonocardiograph can predict it or a predisposing factor. Now, if you've got a shortening second sound, it doesn't mean you're going to have a heart attack. But everybody who has a heart attack does have a short second sound, second period, rest period. Okay, so that's what we look for. So the rest period would be shorter in the person who's got an imminent cardiac failure. So if you do pick something up on a functional test like this, there's plenty you can do. Once you've made your diagnosis of what it is that the patient needs. So we talked about the first sound and the second sound. And now, when the heart contracts, when the ventricles contract, blood is pushed into the arteries, into the aorta and the pulmonary vessels. And as it goes, then the tricuspid and the mitral valve between the ventricle and the atrium closes. And that's the first sound. So the lub is the closing of the valves between the atrium and the ventricles. Okay, that's the first sound. The heart contracts, the valves shut, and then we squeeze blood into the aorta and the pulmonary vessels. And at the end of systole, or contraction of the heart muscles, the aortic semilunar valves, as they're called in the pulmonary, shut, and that's the dub, that's the second sound. Then we have our rest, and then the cycle starts again. So that's our lub-dub. Okay, so let's just remind ourselves about taking the cardiograph. Obviously, if you use a standard stethoscope, you don't need to do this part of the procedure. Okay? So you would just lay the patient down to do the standard stethoscope, and you would have a listen as the doctor or practitioner to the patient's heart, and then when you're clear that you can hear it, you then put the stethoscope on the patient, okay, and the stethoscope hearing part onto the patient's ears, and then you do the muscle test. It's as simple as that. Disadvantage of that is not only do you not get the recordings, but the patient is usually lying down supine, which means the heart tends to drop back. So the sound of the heart is not as clear as when the patient is sitting. And even with some patients sitting, it's not as clear as if you want to drop them forward. So we tend to just listen to the patient with the stethoscope, first of all, and make sure we got the best position to take the recording, and also the loudest position. So if it's not very loud, then we just drop the patient forward a bit and brings the heart a little bit forward, and then you hear it much better. But even so, with some people, it's still very quiet. So I tend to do my phonocardiographs with the recording, with the patient sitting, and we use Erb's point, which is the point on the third costal interspace on the left. So we're just left of the sternum in the third interspace. And the reason for that is it's the ideal position for picking up all four valves. So it's a midpoint, if you like. So instead of listening over the aortic valve or the pulmonary valve, etc., we go straight for the middle bit and then we hear the best of the bunch. But if you wanted to listen specifically at an aorta, then you would go over the aortic valve. And that may be later on when you're a bit more proficient and you're listening for aneurysms, or you suspect there could be an aneurysm, you may want to look just over that one particular area. So we tend to do the recording, if we're doing the electronic one, for 20 or 30 seconds. Now 20 or 30 seconds usually gives you long enough to see abnormal patterns. So far we haven't seen a perfect heart, we've seen a few good ones, although they haven't proved to be perfect. But if you're going to get a bleep electrically, it may be on a regular pattern. Okay, it may be every sort of third beat, every fifth beat, it could be every 20 beats. But so if there's an abnormality in about 20 to 30 seconds, you'll see it. So if there is an abnormality, then just leave it playing or recording for a bit longer, and because you may then start to pick up a pattern. But the majority of people, you see more or less everything that you want to see in 20 to 30 seconds. And here's the ordinary standard stethoscope with Clive being the patient there, and the patient then just holds the stethoscope down, because if you don't hold it down, it'll come off and it won't have the pressure, so the patient won't hear it anymore. So you can use either technique to do the recordings and play it back. Now what we found is that very few people have a so-called perfect cardiograph. A lot of people have goodish ones, and an appalling number have bad ones. And Jill and I were quite amazed. We were at the ICAK conference, and Jill spent most of the 4 days that we were there just doing recordings for the doctors at the conference. And we found very few good ones, very few good ones at all, which was quite surprising, and because they're the healthy people, or supposedly be the healthy people. So when you do this with your patients, don't be at all surprised if you don't get a textbook case come up. So we found that there are variations in the height of the sounds, the first sound and the second sound. They could be smaller or they could be bigger. There's variations in the distance between the resting period and the working period. We talked about imminent cardiovascular problems. There's differences in the sounds before the first sound and the second sound, called murmurs, so we can get stenosis or regurgitation. So we get stenosis before the first sound and regurgitation with the second sound. We had abnormalities in the firing sequence here with bundle branch blocks, where we've had differences in the firing of the bundle of His. So we've had split first sounds here. We've had extra long periods of rest periods here with arrhythmias, etc. So we've seen all sorts of varieties. But in the majority of people, it's a combination we found, and that surprised us. You get a bit of this and a bit of that. And it's very difficult to sort of think, well, yeah, Chris taught us that. Do I give vitamin B1, B2, B3, fatty acids, a bit of heart tissue? The answer is test, isn't it? And this is the simple form. simple way of doing this. So let's take somebody. If I could have a patient, Jill, if you want to get ready. Let's just run through the procedure again. And does anybody, yes, you'd like to? Okay, so if we pop you down here, or if you go and sit with Jill there, we'll need to just disconnect. We'll just run you through the procedure. With the varying programs, whoever you get the stethoscope from, you have to remove the mains. So you need a laptop or an iPad. You can do it on iPhones. We're not that impressed with the results you get on the iPhone. You know, they can be, and if you're out in the field, you know, that might be the only way you can do it. But generally, a laptop is good. Can we get in there now and follow what Jill's doing? Okay, so with a bit of luck, we'll be able to do this live. So can you swap over onto the other computer? So the patient is sitting, perhaps dropped a little bit forward, the stethoscope's turned on. With this one, the ThinkLab one, we turn the amplifier off because it's too powerful otherwise. Jill's going to have a listen with the stethoscope using Herb's point for the best position. The programmer is all ready to go. What she's done sensibly is to put the arrow, the cursor there, right by the on button, and then she's off. And any sound? So we're going to see if we can actually hear this live. Okay, don't worry about it. Okay, so she's done 21 seconds, that's fine. And let's now wind it back to the beginning and plug in the speaker one, where you take the stethoscope out and plug in the speaker, and then we should hear the sound. And what we'll do now, if you put the loop on, can you turn the loop on there? Okay, so now we should be hearing the sound. A bit more volume, Gary. Can we get more volume? Let's really pump it up. Now, because it's looped, at the end of the 20 seconds, it will reconnect. So sometimes, if there's a little aberration at the beginning or the end with a chest sound, you can pick where that is. So that's of no significance, and their brain won't pick that up. But we hear it just as a in between, little hiccup. Okay, now, this is in the 20-second. This is an interesting area here. So you wait for the timeline to come back to here. This is very good. Can we get any more volume up, Gary? Here. Now listen. Did you hear it? Yes. There's a third sound in there. It all happens in 0.8 of a second. It's very, very quick in there. So what you should be able to hear now is lub-dub, lub-dub, lub-dub. So when we look at the graph here, the phonocardiograph, let's just listen to this once more. Okay, so we've actually got a split sound here. So there's nothing abnormal, it's just a split sound. And it's a once-off in the whole process. It's of no significance at all. Okay? If it was every 2 sounds, every 3 sounds, regular basis, then it would be significant. But because it's only a once-off, everybody has something. Okay? Now, if we just put a stop on it. Okay, and let's wind. Can you make sure it's at the beginning? I think that's it. Good. So what we do is, like an X-ray, we have to have a procedure in our mind of how we look and interpret it. So what we do is we say lub-dub, rest, lub-dub, rest. So what we can pick out is either the first sound being twice the size of the second, remember, or 3 times, or the resting period. So we can see beautiful resting periods here. See, twice the distance. Okay, but where's the first sound in relation to the second sound is not very much there at all. Okay, so see that one? That's the first sound, second sound, gap. First sound, second sound, gap. First sound, second sound, gap. And it's a brilliant cardiograph, but there's virtually no first sound. Okay, the lub-dub, lub-dub, lub-dub is more or less the same. It should be lub-dub, lub-dub, lub-dub. So what we've got is a decreased first sound. And this we found Over the 8 months since we did phonocardiography 1, and we were finding this 4 months before that, this is the most common finding that we get, is a decreased first sound. Now the first sound is the closing of the valves between the ventricle and the atrium. Okay? In other words, the mitral valve and the tricuspid valve. This is the shutting, them snapping shut. So if it's a quiet sound, it means there's not enough contraction of the heart. Okay? Now we know that the muscle of the ventricle can be up to 4 centimeters thick. It's a powerful muscle, and it works every 0.8 of a second. And basically what it means, it's not contracting enough, okay, to pump out. Now we can't say it's 95%, we can't say it's 80%, we don't know, but it's not enough for what it should be. And that's the most important thing. So the heart is not working efficiently, which means it won't pump the blood out to the areas where the blood is needed, which can be not only the big toe, but all the organs in the body and the brain. So these people will suffer from a lack of blood to the brain. And we know that the blood not only transports nutrients to help feed the cells, repair the tissues, etc., but it pumps oxygen. Okay? And oxygen, as we'll see, is a vital commodity in every tissue of the body. So these people fundamentally will be suffering from what is colloquially called hypoxia, which means a lack of oxygen to the parts that matter, in other words, to the brain. Now the first vessel off the aorta, which is the major, or the artery coming out of the heart, the first vessel goes to the most important organ in the body, and that's the heart itself. Okay? So the pickings of the blood supply, and the blood coming into the left atrium comes straight from the lungs. So we've got oxygenated blood coming straight into the left atrium, going down to the left ventricle, and the pickings of that enriched oxyhemoglobin, oxygen blood, is to the coronary vessels, which go down and split into left and right vessels and get smaller and smaller until they go down into the capillaries between each myocyte, the muscles, tissues of the cardiac muscle itself. So the reason the heart has the pickings is because it needs the best, because it works harder than any other muscle or any other tissue in the body. Okay? Unfortunately, it has the richest oxygen, which is great for the mitochondrial use, but this does also predispose it to making oxygen radicals. So whenever we've got oxygen or free molecular oxygen around, we have the potential to make reactive oxygen species or free radicals, which generate inflammatory products, as we'll see later on today, which can lead to damage to the coronary vessels. So although it's good, it's got the top quality blood coming in, it also has the top quality risk if we don't have sufficient antioxidants and enzymes to reduce or metabolize these reactive oxygen species. Jill, could you put on that other cardiograph which I did yesterday, just to show you an example there? So if we just shut that one down. So what I'm going to do is I'm going to show you one which I did yesterday. And this would be just your luck. You get yourself a digital stethoscope and you run the first one with the first patient and you get this come up. And it's recorded, just if you shut that down, yeah, save it. We must remember always to save patients, because the good thing is you can bring it back then next time and we can say it is. What I do is I save, put the patient's name, put the date, and then I put on what I put them on. So you might put on B12, B3, zinc, some detoxification, whatever it is, but make a note of it, because then it validates which reading you get. So what we're going to do is draw one up which will be one which might take a bit of interest, and we'll talk about this during the day of what it might be. So now it'll be in the recording. Okay, let's take that. So this is one we did yesterday on somebody who wanted to have a demonstration of what the phonocardiograph does. And what we've found is, when you look at it again, same procedure, where's the first sound, where's the second sound? Here's the first sound, second sound, here's the gap. Okay? So no problems there as far as the rest period. First sound, second sound, gap. And then what happens? So we have a sound of some form come out here, which is a complete split in the bundle branch, the first sound there, and then probably that's the first sound of a small second sound gap. First sound, second sound gap. Probably that's the gap with a little bit first sound, second sound gap, and then we've got this complete split again here. If you can run along there, and we'll see that we've got it there, we've got it over there, you can see. If we run back to the beginning, okay, so what we've got is a regular irregularity, okay, or it's irregularly regular, or regularly irregular. Okay, so in other words, we've got an abnormality which is there a lot. If it was a once-off, it probably was not significant. It could be due to a number of things, but basically what we've got is an electrical problem here with the firing sequence, okay, and it's regular, which means that the AV SA node, the pacemaker, and the AV node are not firing correctly, okay, and what we've got to do is try and work out why that is. Now, I can't tell you, because there's only one person who knows the answer to this, isn't there? And that's their heart or their brain knows the answer to this. What I do know is there's an abnormality in the firing sequence, and that the heart has an inherent rhythm, which we know is set at 72 times a minute. We know this can be overridden and speeded up through the sympathetic nervous system and slowed down through the vagus nerve. So there's influences that can cause abnormalities within our sympathetics and within our parasympathetics, or within the inherent rhythm of the heart. We know that the firing sequence can be upset if we don't get enough oxygen, because the oxygen helps us to make ATP, which is necessary for the firing of the nerves to work correctly. It could be that there's a deficiency of another vital nutrient in making ATP, like magnesium or one of the vitamin Bs. There could be a poison in there, which is having an irritation and causing neurological inhibition. For instance, toxic metals can do this, certain chemicals certainly do this, and risks from radiation. So we've got a holistic approach, functional biochemistry approach. Almost anything can cause it. So we've got to whittle this down. So we can tell by feeding this back to the patient and then going through the different procedures of the eye positions and so on, whittle it down, and then using the knowledge that we've got to find precisely what that patient needs. Okay, so to get in now to the subject today of optimizing human performance is we need adequate amounts of energy. Everything at the end of the day is based on energy. Scientists tell us that nothing is solid in this room at all. When we look down to electron microscopes and smaller and smaller and smaller, even when they look at atoms, there actually is nothing solid in an atom at all. It's all energy spinning round, which gives the appearance of being solid. And we know that mass can be weighed by Einstein's rule, but it can be converted to energy and energy to mass. So energy is therefore important. We know that energy cannot be destroyed, but it can change its form. And when it changes its form, we can measure it. When it doesn't change its form, it's harder to detect. But we can measure certain energy when it becomes heat or when it becomes light. So we can measure energy. And energy requires two vital components. Energy requires the right nutrients or the fuel to go into the burner and the air to burn the fuel. So if you imagine getting enough wood to your stove in your kitchen, or let's say a wood burning stove these days, okay, you've got to have the wood to be able to put into the burner. But at the same time, you've got to have the air. So if the damper is in the burner and there's not enough air circulating, the wood won't burn. Right. Or it'll only partially burn and smoke a lot. So that's an important part of it as well. So we need the fuel and we need the air. We need no toxins or less amount of toxins that we can, because we know that toxins inhibit almost every enzyme in the energy production. Both glycolysis and the Krebs cycle and in the mitochondria are affected by toxins. There are very specific toxins now which are known for different pathways there. We don't want infections because we know not so much the infection increases the heart rate and has an effect, but it produces a lot of toxins. So we have endotoxins produced inside the body as toxic byproducts from bacteria. We know all about parasites, with their different ammonias that we produce, which are very toxic, phenol, toluene, byproducts from parasitic metabolism. We know about fungi, and fungi are big issues right now in this part of the country, and that fungi produce acetaldehyde, acetic acids, and ethanol. But some fungi, particularly the pathological ones, produce methanol, and not ethanol. So not only do you feel blind drunk, but you feel blind drunk and toxic, like a meth drinker, which then goes on to make formaldehyde rather than acetaldehyde. And formaldehyde can be pretty, again, neurotoxic and certainly nephrotoxic to the kidneys, and therefore will affect the heart. Why did I say that about round here, this part of the country? Is because of all the rain we had in the winter. The ground is super saturated. The surface is as dry as a bone. Everybody's going out there with a hose pipe. But underneath, it is as wet as can be. It was only 4 months ago the ground was underwater everywhere. That hasn't disappeared too far down. So everything is very damp still. It's humid, it's damp. This is ideal mold growing. A piece of bread, wholemeal bread, which we make, Jill makes up, is got a mold on in 2 to 3 days in the kitchen, and that's in a new house. This is amazing, just the molds that are around. And they're talking about, so many people have got hay fever this year. They haven't got hay fever, they got mold allergies. This happened in New Orleans some years ago after the hurricane that they had there. People couldn't go back to New Orleans because it was so moldy. The houses hadn't been lived in and the molds were enormous, and people were getting really chronic chest problems. And this is what this sneezing is at the moment. It's the molds. The molds are going to be around for a long time until everything really dries out. So absence of infections. And of course, a positive state, a mental state, emotional state is very important for optimizing performance. We all saw what happened to Andy Murray. Now, anybody hazard a guess as to guy wins every single set and then loses every single set in the vital match? Why? What happened? It doesn't take too much to add things together, does it? In my opinion, anyway, what happened? The girlfriend is one part, the mother's over there, and the trainer's here. Why? Okay, say no more. So the positive attitude had gone from him. He was winning every set before, every set, because of his emotional state. His emotional state had changed. And it was very, very unfortunate, that happened.